The human papillomavirus is a group of viruses that are very common across the world, mainly transmitted via sexual contact. Whilst most HPV infections do not cause any problems, some types such as HPV-16 and HPV-18 are classed as high risk and cause approximately 5% of human cancers. Of the cancers caused by HPV, cervical cancer is the most prevalent, with 70% of cervical cancer cases and precancerous cervical lesions being attributed to HPV-16 and HPV-18 infections. Cervical cancer is the fourth most common cancer amongst women globally, with approximately 570,000 new cases in 2018. Each year, cervical cancer cases cause approximately 250,000 deaths. This video will explain about HPV-16 and its role in causing cervical cancer through the interaction with a tumour suppressor gene called p53. Papillomaviruses are non-enveloped icosahedral DNA viruses consisting of a circular double-stranded DNA genome surrounded by a simple capsid. It is a small virus with a diameter of approximately 55 nanometers. Early genes expressed by papillomaviruses, including E6 and E7, act as oncogenes that promote tumour growth and malignant transformation. HPV-16 has two virally encoded structural proteins, the L1 major capsid protein and the L2 minor capsid protein. Here we see the asymmetric unit of HPV-16L1, which is the smallest part of a crystal structure to which symmetry can be applied in order to generate the complete virus. This asymmetric unit assembles to form an icosahedral virus with various symmetry. There are 360 copies of the L1 capsid protein in the papillomavirus. HPV-16 causes cancer by integrating its genome into nuclear DNA. When the viral DNA is inserted, the new DNA that is replicated from the nuclear DNA will have the viral genes inserted. This means that all new copies of DNA will contain the cancer-causing code. Once the viral DNA is embedded and replicated into the host cell, various cancer-causing genes are also encoded, including the E6 oncogene. This oncogene produces an oncoprotein, also called E6, which binds and inactivates p53. p53 is a tumour suppressor gene which plays a key role in regulation of cell proliferation. In most cancers, the function of p53 is lost, allowing cancerous cells to multiply and form tumours. Here we can see p53 binding to the binding cleft on E6, causing degradation. This degradation then prevents p53 from inducing either the growth arrest or apoptosis of infected cells, leading to uncontrolled cell division and evasion of cell death. There are two stages of prevention against cervical cancer before treatment of invasive cervical cancer. Primary prevention is vaccination against HPV, and secondary prevention is screening and precancerous lesion treatment. Vaccines protecting against HPV-16 and HPV-18 are recommended and have been approved in many countries, including the UK, with clinical trials and surveillance showing that HPV vaccines are safe and effective in preventing HPV infections. In conclusion, E6 is a central oncoprotein driving HPV-mediated cancer. Therefore, it presents the most effective target for therapy. Current strategies targeting E6 include vaccines, T-cell therapy, genome editing technologies, and immunotherapeutic targeting. Future therapeutic suggestions include mutagenesis, i.e. changing, of critical positions at the interfaces where p53 binds, therefore blocking p53 binding and ensuring that p53 does not lose its tumour suppressive function.